Typically, DNA is on linear strands, so you have 46 chromosomes, and each of those 46 chromosomes is a linear strand of DNA. However, in some cancer cells, what we found is that there are small pieces of circular DNA, and these are called extrachromosomal circular DNA amplifications, or ecDNA in short. The presence of ecDNA enables cancer cells, such as glioblastoma cells or brain cancer cells, to adjust to challenges in their microenvironment. For example, the presence of ecDNA enables those cancer cells to deal with the toxic effects of radiation therapy or chemotherapy or any other therapy for that matter. Our lab is a brain cancer lab. We are dedicated to developing therapies to better treat glioblastoma and glioma patients. Glioblastoma is a grade 4 brain tumor. It's the most common malignant brain tumor in adult patients. It's a devastating tumor type. Patients that are diagnosed with this kind of cancer often have a very short lifespan following diagnosis. This is a tumor type that really has not seen any progress in clinical treatments in the past 30, 40 years, and we're very motivated to change that. My lab has traditionally done a lot of profiling, which means that we take a tumor sample from a patient and generate lots of data from this single tumor, sometimes billions of data points. And then we use computational data science approaches to make sense of those data sets. Now we are in a phase where we're trying to take what we've learned and do functional experiments, and where we can test hypotheses that are derived from our data science analyses and turn those into targets. And once we have targets, which is the stage where we are now, we have targets, we want to turn that into, can we now develop small molecules or other treatment modalities that aim for those targets? Anybody who has had cancer or who knows somebody that has been treated for cancer knows that the side effects are often very challenging and sometimes lead the patient to no longer be able to take a certain therapy. For example, many chemotherapies are so toxic, the patient gets so sick that treatment has to be discontinued. So targeted therapy is a way to treat patients with very high efficiency. You're really trying to only hit the cancer cells and leave the other cells in your body alone, and thereby minimizing all those toxic side effects. Our strong motivation is to develop these molecules, make them safe to use in humans, and then put them into phase 0, 1, 2, 3 trials, and ultimately approval by the FDA. I work closely with varieties in neuro-oncology and neurosurgery and pathology and radiology. Glioblastoma is a disease that no individual is ever going to cure. This really has to come from collaboration, open science, to together do better for these patients.